And our next speaker is Vittorio Sebastiano from Turn.bio.
First of all, I would like to thank Morten, Alex, Daniela, the organizers. It's really a great pleasure to be here today, to be among such an incredible group of people that are working in longevity. Today I will talk about the recent progress that Turn.bio, the company that I co-founded with Marco Quarta, is doing in the context of epigenetic reprogramming as a technology to rejuvenate cells and tissue. Which is a bit in line with what David Sinclair said yesterday. Since I want to cover as much data as I can, I want to get straight to the point. So like David, I am a firm believer that actually the fundamental linchpin of aging at the cellular level and of course then at the tissue and organismal level is the phenomenon that we call epigenetic drift, which is a progressive accumulation of epigenetic mistakes, errors in the epigenome that progressively lead to the loss of function in cells. So here's a very, very kind of simplistic representation of this concept. So you go from a young cell where you have a fully functional epigenetic program, and then with time, in part because of a developmental program, which is species-specific, and also in part by environmental factors, diet, sleep, stress, and so on, this epigenetic change becomes dysfunctional. And so genes that are supposed to be off get turned on, genes that are supposed to be on get turned off. This has profound implications because since the epigenome is really the fundamental program that regulates every single function in the cells, by changing the epigenome, you impact basically pretty much all the so-called hallmarks of aging. Now, the good news, though, is that the epigenetic program, unlike the genetic program, is an analog program, and it can be reversed. It can be rewiped, reset. You can choose the term that you like. But how? Well, when I started thinking about this problem, thanks to the fact that I am a developmental biologist and a reproductive biologist by training, I really, I had an epiphany, and it was like, well, I realized that nature had already figured out the perfect way, actually, to prevent aging from happening, which is reproduction. If you think about that, we, as individuals of the human species, we age, but the human species doesn't age as a whole. Life doesn't age as a whole, and we are, philosophically connected with the first life that occurred billions of years ago on this planet. How? Through reproduction. So the secret is there. The solution is already there. We just need to understand it, and we need to hijack those processes, those principles, and apply them to cells that are non-germ cells, that are non-embryonic. And so the inspiration really came from the seminal work of these two individuals, John Gurdon and Yamanaka, who in different ways, different systems showed that this reprogramming to an embryonic cell is possible. And you can turn any cell into an embryonic-like cell or an embryonic cell, through somatic cell nuclear transfer in the case of John Gurdon, or with IPS technologies in the case of Yamanaka. And this is phenomenal. I mean, I don't have to explain why this is so important. But what they did not realize right away was the fact, because this was historically put in the context of, can we generate patient-specific embryonic-like cells so that we can generate new organs? What they did not realize right away was the fact that as you do this change, so you turn a somatic cell, a skin cell, into an embryonic-like cell, you're also making that aged cell that you start with into a very youthful cell, which is the embryonic-like cell. So in 2014, we had this idea, and we said, well, wait a minute. If you can make a cell that is very youthful, pretty much age 0, what if by tweaking and by really controlling this process of reprogramming, what if we can just change the age of the cell without changing the identity of the cells? Because we don't want to make an embryonic cell, Because, well, yes, we do, but making an embryonic cell means also that you have to differentiate then the cells back into whatever, liver, brain, and so on, which is not easy. So we said, well, what if we just partially reprogram the cells and we make that cell, the same cell, but younger? And that's what partial reprogramming or transient reprogramming is all about. We call it ERA, Epigenetic Reprogramming of Aging, but that's really the idea. The idea is really to transiently reprogram the cells so that you don't change their identity, but you make them more youthful. And so to make a very long story short, we published the foundational work in 2020, and now using a cocktail of 6 factors and using mRNAs. And now, Turn is trying really to bring this into clinical fruition. And I'm very pleased to say that actually after our work and, Belmonte's work, he published first in progeroid mice. We were the first to publish in human cells. After those 2 publications, there has been really an explosion, and the field is booming. And now there is really a lot of evidence in vivo that this technology works. It works in mice, it works in rats, it works in non-human primates, and it works across different tissues: muscle, skin, hippocampus, blood cells, you name it. So it's a very reproducible and robust technology. But how can you translate that POC, proof of concept, into something that can be used for people? And so, again, this is what Turn is all about. So we are trying basically to develop these, to harness the power and the potential of this technology and really try to make something that could be used to impact the health span of elderly people. And basically, to summarize, what we do is really to try to put together 3 elements. One is the biological foundation, which is the concept that the epigenetic information of aging can be reset through transient reprogramming. And the second is the how, which is the cargo. We use mRNAs, and the delivery system, which is the liquid nanoparticles that we need to basically make sure that these factors are delivered into the cells. And this is, we chose the mRNAs for a variety of different reasons. The most important one is that it's by far the safest approach because it doesn't RNAs do not integrate into the genome, so there is no insertion of mutagenesis. We can very tightly and finely regulate the expression of each of the 6 factors. And I just want to make a clarification here because otherwise the wrong message comes across. It is not true, it's an inaccurate oversimplification to say that if you express MYC together or other factors together with the Yamanaka factors, that those are toxic to the cells. It's not necessarily true. They are toxic if you don't know how to express them and if you don't know how to tightly regulate the expression. MYC has a very important, for example, repressive activity on retrotransposable elements. It's involved in mitogenesis. So if you can just pulse it for a very short time, it has a lot of beneficial effects together with the other factors. So I think this needs to be clear because there is a lot of misconception out there, which is driven by the fact that people are using, in my opinion, models that are not the right ones. For example, DOX-inducible transgenic, mice or viral vectors that are very tricky when it comes to regulating finally the expression of these factors. Okay, so having said that, this technology can potentially be applied to a variety of different tissues. We have plenty of data in ophthalmology, muscle, but for the sake of today's talk, I'm going to just show you what we have done, what we are doing in dermatology and immunology. So to come to dermatology, which is our first in vivo pipeline of ERA, dermatology is divided in 3 layers. So we do primary cell work. And I want to clarify, we work only with human cells and human tissues, okay? Because we want to make sure that what we see is really translatable into people. So here you see an example of how on primary cells in fibroblasts, ERA, which is this transient reprogramming, results in a broad transcriptomic resetting of the cells globally. And if you just dig into the data, you see that, and you do, for example, gene ontology analysis, you see that the genes that are implicated in this reprogramming, or the genes that are activated in response to transient reprogramming, are implicated with ECM secretion and organization, collagen-containing ECM again, PDGF binding, cytokine-mediated signaling. We typically see low pro-inflammatory cytokines and high anti-inflammatory cytokines expression, and so on and so forth. Then what we do is to, and if you dig into the data, you see that if you just look at fibroblast-specific genes, you see that there is quite a remarkable change in the expression of fibroblast-specific genes. And you see that genes that go down with age come up with age, with the reprogramming again, for example, all the collagens and the extracellular matrix proteins. And the bad guys, the ones that are not supposed to be expressed, but they are expressed with age, get downregulated, for example, the MMPs. So, and here you see, so that's true at the RNA level, but at the protein level is also true. So, for example, we see downregulation of SOD2 and MMP1. And then you see a number of other markers going down, MMP9, P16, and an upregulation of H3K9 trimethylation, which is a youthful marker, epigenetic marker. So then the next step is, okay, so now how do we go to the next level of complexity? So what we do is we isolate human skin from people, from plastic surgery samples. And what we do, we make skin biopsies that we keep in culture, and then at the same time we screen a delivery solution. So we have a proprietary way to actually develop liquid nanoparticles for the delivery inside the cells. And you have seen the movie on the side, so we can screen rapidly the best formulation that gives the highest transfection efficiency with the lowest cytotoxicity. And when you combine those two things, so when you now go into inject era, we try to measure efficacy. And so first, before measuring efficacy, we need to really rely on stable biomarkers or reliable biomarkers of aging. So in this case, we have built our own using skin biopsies from different age groups. We have seen that there are markers that go up or go down with age. So those are perfect markers to look at. And after ERA treatment, we see that, for example, SOD that goes up with age goes down with the treatment. So the samples are less prone to oxidative stress. And the markers that go down with age, for example, collagen 7, go up again after the treatment in the tissue in culture. The next level of complexity now is, well, now we want to do this in a living human skin. So obviously we cannot directly inject this into people as of yet. So what we have to, we spent a lot of time developing an in vivo model of skin. So we take the skin now, instead of putting it in culture, we put it on the back of an immunocompromised mouse so that it stays alive for a long time. And on the right, you see how then we microinject the LMPs with the ERA factors inside subcutaneously. You see that kind of bulging thing over there on the back of the skin. And the mice are perfectly fine. Sorry, this was supposed to start on its own, but it's not okay. And you see that the mice are perfectly fine, and the skin survives on the grafts. And here you see data up to basically 6 weeks post-transplantation. You see that the skin is viable, so it grafts. And so now we can really use this model to show in vivo efficacy proof of concept. So this is work in progress. So hopefully, maybe next year, I'm gonna share some data on this, or hopefully sooner than that. So now I want to very quickly switch gear. So now you see how ERA can be used for tissue regeneration in vivo, and then also how it can be used ex vivo for cell rejuvenation. And the pipeline that we are looking at is immunology for a very obvious reason, because as we know, our immune system ages with time, and the immune system is really important for a variety of different things, and it would be important to rejuvenate it. Also because this could be really the first systemic approach Not in the sense that we are trying to target every cell in the body. I don't know if we will ever able to do that. But we are targeting the cells that go systemically everywhere in the body. So this could be really a systemic solution of rejuvenation by rejuvenating the cells that go everywhere in the body. And so, you know, immune senescence results in a variety of different things, but very quickly I want to show you that ERA in this case, so we're treating T cells that are coming from old donors. In this case, you see again that after the treatment, which is approximately 3 days, there is no change in cell identity. So this is a very important concept I want you to go with, to live with today. We are not changing cell identity, and we are confirming it across a variety of different markers of T cells. So the cells are still the same cells, but they are much more proliferative when you engage these T cells with tumor cells that are capable, that the T cells are capable to recognize and kill. So they over-proliferate, so they're much more fit and, you know, to respond to the tumor environment, as you can see here. They express a much lower level of exhaustion markers. So that means the cells, when they see the cancer cells, enter an aging-like phenotype because the cancer is signaling, well, leave me alone. So just, you know, shut down, leave me alone. So those are cytokines that actually age the T cells in a way. We see that those markers of aging are actually, you know, dampened in the treated cells. So the cells are much more robust in response to the tumor cells. This results in a higher killing capacity. And so I want to really leave you guys with just, you know, this visual proof of what is going on. There's something wrong with the movie. Let me see. Okay, so you see here, in both left and right are pretty much the same thing. So the green cells are the cancer cells, so the cells that the T cells are supposed to be killing. And the non-green cells are the T cells that we are adding as killers. You see that in the control situation, the cancer cells at some point overtake the whole you know, system, and they are and the T cells are no longer capable of killing them because the cancer cells are just more, you know, potent. On the right side, you see instead the ERA-treated cells, so the same cells that are treated, they are much more fit and more robust, and they can very effectively kill the tumor cells and get rid of them in a much faster time. And so I want to conclude here and just reiterate the idea that, you know, these can potentially be applied to a variety of different tissues. We have plenty of data in muscle, in ophthalmology, in osteoarthritis, in blood cells, in skin. But, you know, these two are the first lead programs. And I want to conclude by thanking, you know, the group, the leadership of Turn. Obviously, I didn't have you know, we're 30 people, so really the ones that I need to thank are the people that are doing you know, the experiments in the lab, the investors. Marie-Louise is actually here, and I just want, as I take maybe 1 or 2 questions, I want to leave with this that summarizes what, in a much nicer way, you know, what we're trying to do. Thank you.
Thank you so much for a great talk. We have time for a question.
Vittorio, so terrific on so many levels, and in particular that you decided that you'll invest in treatment and not only in the discovery, and really best wishes to you. You know, the mRNA is of course a great technology, but you cannot really deliver it to every organ for now. So I thought the skin, I thought you're going to tell us there's a cream of mRNA, but no, it's a transplant. So I don't understand the clinical indication here. Can you, because it's relevant also to some of the other indications. How are you thinking to do that?
Good point, Nir. Well, there is not a cream yet. One day there will be probably a cream. Obviously, you need also to kind of cope with how far the technology is, you know, the surrounding technology is also developed. So right now, the first route of administration is going to be microneedling or microinjection to basically physically cross the barrier and deliver the RNAs and the lipid nanoparticles, you know, in the dermis. So that's going to be the solution 1. But we are also thinking about potentially developing a topical solution that could, on its own, go through the barrier and deliver. You're absolutely right. Delivery is the #1 challenge for RNAs, absolutely. At the same time, I think that there's a lot of things changing, quote unquote, thanks to what happened with COVID. We have realized the power of RNA technology thanks to the vaccines, right? And now we are understanding that there is a need for gene therapy, for example. There is a need to really crack the code of delivery and really develop solutions. And that's also what we are heavily working on. Because, yes, in the case of vaccine, it's enough to deliver maybe to 5% of the cells. In the case of this, we have to deliver in a very high number of cells. So we have to find, the breakthrough solution for that. But I'm confident that many will find solutions because there's a lot of people working on it, and there's going to be a lot of changes in the future.
Thanks. Thank you again for joining and also for sponsoring the conference.
Thank you.